In our last video, we talked about what enantiomers were and the presence of chiral centers. So remembering that enantiomers are isomers of the same molecule that have a different spatial arrangement in 3 dimensions, and therefore they rotate plane polarized light in different directions. So basically these are compounds that contain chiral carbons, so they have a chiral center, and therefore they will interact differently in 3 dimensional space in binding reactions such as with receptors and enzymes in our body. So we're going to have a look now at what that means for drug action and how different compounds react as drugs when they're different enantiomers. So essentially, it's important to realize that chiral compounds are very common in nature. When something is synthesized in the body, it is synthesized using enzymes. Because of their specific binding sites and the nature of the way that enzymes and receptors work, they often produce chiral compounds. So the way that the groups are attached in 3 dimensions are very specific as well. So the majority of naturally occurring compounds used as drugs or pharmaceuticals are extracted as single enantiomers. So we don't tend to use pharmaceutical products which are a mixture of enantiomers. We tend to use a single enantiomer. When we synthesize these in the lab, chiral compounds are generally formed as a racemic mixture. Because in a reaction flask, we don't have the ability to control the 3 dimensional environment anywhere near as easily or as well as what an enzyme does. When we make them, they tend to be a mixture of both of them, and then we need to find ways to purify these, which can be quite challenging due to the fact that the normal methods that we use to separate mixtures of compounds won't work with enantiomers because they are chemically the same and physically the same, except for that 3 dimensional aspect. Generally, when we're talking about pharmaceuticals, only one of the 2 isomers will be what we call pharmacologically active, that is, will have the desired therapeutic effect. The other isomer may just be inert, i.e. it does nothing when it binds, or the worst case scenario, it can cause detrimental side effects, as we saw with the thalidomide. So it can be considered acceptable for a racemic mixture to be marketed as long as the non-therapeutic isomer, the one that doesn't have the effect that we want, is inactive or its side effect is non-detrimental. So that's another aspect that needs to be tested when we're looking at drug trials and drug testing is the compound racemic? Will it need to be purified from that mixture, or can it be produced as a racemic mixture? Generally, there's been more scrutiny placed on the use of racemic mixtures since the thalidomide tragedy and our better understanding of what different enantiomers can do in the body. So this works essentially by the fact if we look at these 2 enantiomers here, okay, if we imagine a mirror here, we can see that these are a mirror image of each other. When it comes to the drug receptor site, only one enantiomer will fit in exactly to match the binding of the enzyme or the receptor. So the correct enantiomer will fit like a lock into a key, whereas the incorrect enantiomer doesn't fit quite as well. It may have some bonding interactions, so this is how we can see that it elicits some effect, but the interactions aren't exact, they're not specific, so it can result in no medicinal effect at all or a detrimental effect. We've talked about thalidomide a lot since starting the option, and this is one case study where we can look at the impact of enantiomers. Okay, so if we look at this enantiomer here, so where we have the functional groups 1, 2, 3, 4, so we have a chiral carbon in the center here, this can fit well into the active site of the enzyme. If we swap these 2 groups here, 2 and 3, now what we can see is that if this is my enzyme pocket, if we have 3, it won't have the correct interaction to this pocket, and 2 won't have the correct interaction with this pocket. Therefore, it accumulates in the body. It's not broken down by the enzyme. It accumulates in the body and therefore causes the detrimental effect. This is the 2 isomers of thalidomide here, and this difference is shown simply through, this is our chiral carbon here. If we do the mirror image of this carbon, then this is the one that causes birth defects. So it can be as simple a change as that can drastically change the way that drugs work in our body. So there are a number of other enantiomeric pharmaceuticals, ibuprofen, Nurofen being one of them, where one enantiomer in this case is more effective than the other. Okay, so the other one doesn't have as strong a pain reducing effect. Okay, and that is on this chiral carbon here, where we can see again they're flipped around a mirror image. This one is up, and this ring system coming off here. The ibuprofen, so the one that rotates light to the right, has no side effects but isn't as effective, whereas this S enantiomer, the one that rotates light to the left, okay, reduces fever and acts as an analgesic, mild analgesic at the site of the pain. Okay, other drugs such as dopa, which has 2 forms, L-dopa and D-dopa. Okay, you may have heard of L-dopa in terms of treatment for Parkinson's. Okay, L-dopa is the effective one, whereas D-dopa has no physiological effect. Fluoxetine, morphine, codeine, heroin, all of these contain chiral centers. In fact, morphine, codeine, and heroin have 5 chiral centers. Only one of the enantiomers, only one combination of those 5 chiral centers actually has the analgesic properties. The other ones don't bind into the opioid receptor in the brain in the same way, so they don't elicit the same effect due to them not matching that specific 3-dimensional arrangement. So when we think about chirality in terms of drug development, okay, because we're dealing with 3-dimensional specificity of enzymes, okay, it really highlights the chirality aspects when we come to drug design. We need to make sure, are we making something that is one enantiomer or a mixture? Okay, when we manufacture drugs, we always get a mixture, so each enantiomer needs to be tested separately to ascertain if it has side effects or physiological activity. Okay, if one enantiomer is active, okay, and it's expensive, then it can drive up the price, and especially if we have to deliver it as a single isomer rather than the racemic mixture. Okay, because these things increase the chance of unwanted side effects, because we can have, even if it doesn't have a therapeutic response or a side effects, because it's not being broken down in the same way, because it can't bind into the enzyme, we may see accumulation of that compound in the body, and that accumulation may become toxic in and of itself. So the way we deal with this is we synthesize specific isomers through the use of chiral auxiliaries, and we'll look at that in our next video.